Hello, everyone, and welcome back to the Future Food Tech monthly webinar series. I'm Oliver Katz, the conference producer, and as the current pandemic continues to disrupt plans across the world and forces many of us to socially distance, here at Rethink, we still want to give a platform to leading innovators to share their groundbreaking insights, challenge the status quo, and connect the wider food ecosystem together. Now more than ever, it is vital that we continue to collaborate towards a stronger, more sustainable and nutritious future. So today, we have invited 3 pioneering startups, BCD Bioscience, Stratuscent and TurtleTree Labs, to present their solutions that range from prebiotic and functional carbohydrates, AI-driven scent digitization, and cell-based clean milk, to a panel of furious sharks. I'm joking. The investors on our shark panel are as brilliant as they are lovely, and we're delighted to have them taking part as well. So we have Ella, the founder of 50 Years, and her co-host Holly as a partner from Joyance Ventures. So a very warm welcome to everyone involved. In terms of the schedule, each startup will pitch their solutions in a 7-minute window, followed by questions posed to each of them from the sharks. After all the pitches are then concluded, Ella will then direct audience questions to the startups and also answer some herself. So please post your questions in the Ask a Question section, which is located at the bottom right of your screen. And also indicate who that question is being directed to. You can also upvote these questions, so the more upvoted the question, the more likely that will be asked. So as always, please feel free to use that comment section as well to virtually welcome our speakers up to the stage, or simply clap and whoop in the comfort of your own home. And if you do experience any technical issues, please comment on the chat as well, and one of my team will be able to assist you. So without further ado, I'm now going to hand over to Ella and Holly for a few words, welcome words, and then lead the webinar through. So thank you very much, both of you.
Thanks, Holly. Good morning, everyone. I mean, I don't know what time what time zone are you in, but it's early morning in California, and I'm excited to be here with you today, and I'm very excited to hear from the 3 impressive startup founders and operators. So my name is Ella Made. I'm one of the founding partners of a venture capital firm, 50 Years. We've been around for 5 years. We back solutions to the world's biggest problems. Food and food tech is one of the big areas in our investing. We have partnered with 9 food tech companies to date, and cannot wait to partner with more. And my colleague here today is Holly Jacobus, and I will give it up to her to introduce herself.
Thanks, Ella. Yes, my name is Holly Jacobus. I am a partner at Joyance Partners. We're a global VC fund focused on companies that create delightful moments. We've actually been around since 2009 is when I got started. And most recently, we've been working on the intersection of health and happiness. We're inception investors, so we usually come in quite early. And I personally focus on food and sort of the happiness sectors in general.
Stephen from Stephen Watkins, the CEO of BCD Bioscience, is there and ready. We can.
I need to be invited into the room.
Great. Okay. Nice to see you. I'm sure everyone is also extremely excited to hear you.
All right. Can everyone see and hear me? Great. Okay. So at BCD, we're looking at the world of carbohydrates and the vast diversity of structures in carbohydrates in the natural world for opportunities to develop new prebiotics, symbiotics, and other therapies for human and animal health. And the way we're doing this is by looking across the natural world for diversity in the chemistry and structures of carbohydrates in foods and natural products, and then applying our proprietary what we call depolymerization chemistry, which allows us to take those natural products and break long polysaccharides down into small bioactive units called oligosaccharides, and then test those oligosaccharides on our platforms for looking at selective microbiome and immune modulation to link those natural sources and their resulting oligosaccharides to human and animal health applications. We're a spin-out of UC Davis, the result of work for 20 years by 3 professors who have worked together and their star graduate student, Matt Amakuchi. So Carlito Labria is a world-renowned carbohydrate chemist. David Mills is similarly a world-renowned microbiologist. Bruce German, a generalist nutrition specialist, and they've worked both academically and in founding companies for the last 20 years. They're the founders of Evolve Bioscience, and this is largely based on technology and patents that they developed at the university, which we've exclusively licensed. And I'm happy to say we actually closed our first institutional round of financing last week, led by Venture Partners, Acre Venture Partners, and iSelect Fund participated in the investment as well. So carbohydrates, it's a little bit surprising actually, given that they're the most abundant biomolecules on the planet, and really the biggest driver of changes in the gut microbiome. But we have very little understanding of the chemistry and the diversity of the chemistry that's present in carbohydrates. The structures aren't described in great detail, and we certainly can't map this diversity of carbohydrate chemistry to the specific foods in which those carbohydrates are present. And so we tend to talk about carbohydrates as fiber and sugar in very generalistic terms, rather than as a specific kind of chemical structure that can be leveraged for specific health opportunities. So we know there are a lot of opportunities for carbohydrates in human and animal health once we can identify those structures, catalog them, and begin to map them specifically to structure-function relationships for modulating the microbiome and other health applications. The technology at BCD really falls into 3 kind of categories. I'll walk through each of these more specifically, but the first is our carbohydrate encyclopedia, and this is the way in which we identify source materials that have interesting carbohydrates for further processing into our glycan library. And the glycan library is a physical library of oligosaccharides, short carbohydrates that we then use to test in our functional assays for specific functionality, whether that be modulating the microbiome, whether it be protecting immune system from activation by agonists or other kinds of insults. This is the way in which we begin to ascribe function to the specific glycan structures that we produce. So in terms of the carbohydrate encyclopedia, the process involves us actually really building a landscape map. In essence, we've profiled about 2,000 natural products with our mass spectrometry techniques. These include everything from a wide variety of plants and yeast and algae and other natural materials, with a focus on waste streams and other natural product streams that are available and can be built into products of increased value. And these mass spectrometry techniques are really novel and actually very powerful. This is work that Carlito Labria and Matt Amakuchi have developed over the last 10 years to look at not only the monosaccharide composition of carbohydrates, but also the specific linkages amongst those monosaccharides that provide the very unique structures of carbohydrates. And from that, we identify a huge variety of these carbohydrate structures, which is shown in the middle picture. And then we can begin to understand the kind of higher order structure of all of these carbohydrate molecules and the diversity by mapping by those structures and looking for similarities amongst natural products. And you can see on the right what we call a phylloglycomic tree. There's a lot of diversity in structures in the natural world, but a lot of similarity also amongst certain kinds of natural products. So this allows us to select natural products with a large degree of variation of chemical structures to test going forward. The production of glycans is really the core technology and the basis of the exclusive license we have with the University of California. And this is really a food-grade process that transforms insoluble and natural polysaccharides into oligosaccharides. And it's a universally active process. So we can take any polysaccharide, we can break it down into short-chain oligosaccharides, which share structural similarity then obviously to the parent natural polysaccharide, and which can be further fractionated if needed for different applications. The functional testing of glycans, we take this diverse collection of glycans we've produced, we bring them into bacterial growth and taxonomy analyses. So we test these glycan structures against single strain microbial cultures, against platforms that allow us to test them in complex communities. We look for changes in taxa as a result. And because of our experience with these mass spectrometry techniques and other functional assays, we're able then to actually take the products of these growth analyses and evaluate them for metabolic output of the bacteria as well as the consumption of the glycans that we added. So this allows us to see not only which specific oligosaccharides and glycans can support the growth of beneficial bacteria, which ones are effective at changing taxonomy of the gut microflora in a beneficial way, but also to look at what the metabolic output of that change is, and where we can find opportunities for those specific attributes. So we're excited about BCD in terms of the range of opportunities that this technology brings us. We think it ranges everywhere from minimally processing natural polysaccharides to solubilize them, bring them back into foods, upcycling of waste streams back into food products, to more significant processing of fibers to create crude and enriched oligopools for selective prebiotics or improved symbiotic formulations, all the way to the identification of very specific oligosaccharides for medical and pharmaceutical applications. And so we're excited to get started here with our new funding and grow the company here over the next year or two. Thank you.
Thank you for the, can you hear me?
Yep.
Thank you for the presentation, and congrats on your recent round of funding. That must be a relief, and I'm very happy for you. So, how expensive is this process to build a really sizable library? I can imagine that, doing GCMS and then like doing the bacterial growth panels and like cataloging everything. How big is, given the cost of the process, how big do you think is the library you have to build to start really see some interesting targets? To start seeing
yes, that's sort of two questions. The process itself is quite economical. We think actually cheaper than existing strategies for generating oligosaccharides through enzymes, et cetera. So we have, we certainly are going to be exploring scale up this year, but we have every indication that this is a very economical process. Cheap reagents, food grade reagents, most production facilities have the equipment. The research itself is more expensive, you're right. So obviously running a mass spectrometry lab and running a screening assay, but that's really as part of the discovery process to bring lead candidates forward. And so, we have about 50 pools right now that we're excited about, and we're planning on generating another 50 this year. So yes, you're right. The research part's a little more expensive, but the end production of the compounds is very economical.
Great, thank you. You actually answered the other question I had about kind of the economics of the oxidative process. Great.
Yeah,
I want to make sure I give it up to. We'll have more questions at the end. Holly, I think you're up next.
Yeah, I'm just curious, kind of who the target customer is here, who ultimately buys this, and how you make money on it, and also what the IP situation is.
IP. So we have existing intellectual property both on the production process, so the ability to actually create oligosaccharides from source materials, and also on composition of matter for the oligosaccharides that we generate with the process. So we feel we're in a very strong intellectual property position. I think we're all by nature food oriented, and so we think of foods first. We're beginning to build out a team that can bring some perspective on higher margin kind of opportunities around pharmaceutical applications, medical food applications. But certainly our hearts have always been in thinking about better food ingredients, both in terms of upcycling waste streams back into foods, so solubilizing fibers and those kinds of things, as well as functional ingredients for foods, better prebiotics, more selective prebiotics for beneficial bacteria.
Got it. Just to clarify, so when you talk about the food, I understand the sort of pharmaceutical industry, obviously, and how that fits. For food, then, is the buyer in this situation like a Tyson or a Procter & Gamble or something like that? Or would it be more the manufacturer?
Yeah, so the distribution is a separate question. I mean, the ultimate buyer is a food company, a consumer packaged goods, food producer. Whether we would supply them directly or work through an intermediary is a different question. But yeah, the ultimate buyer would be a formulator of ingredients for a food product.
Got it. Thank you.
Yeah.
Let's see, I've actually forgot to time that part. I think we're getting close to our 8 minutes, so we should probably move on. Ella, do you have any more questions before we move on?
No, I think there's going to be more questions, time for questions later on. Let's move on, and we'll see how much time we have when everyone is done. So thank you so much again, Steve. This is fascinating, and congratulations on the progress.
Thank you.
And it looks like we've got Fengru on here as well. So I'm going to go ahead and pass the mic over to Fengru from TurtleTree Labs.
Hi, everyone. Can everyone hear me now?
Yes.
So, hi again. My name is Fengru, and I'm the CEO of TurtleTree Labs. We are a biotech company based in Singapore and San Francisco, and we're the first company in the world producing cell-based milk for the future. What we call Milk 1.0 was how we used to traditionally source milk from factory farms, which have been turned into cheese, butter, and yogurt. We know this is unsustainable, and to feed another billion people in this planet, we have to come up with better solutions. So what we call Milk 2.0 has been a shift to plant-based milk like almond milk, soy, and oat. This has been great, but overall still less than 1% market penetration. TurtleTree Labs have been working on what we call Milk 3.0, using biotech to create real functional milk that Milk 1.0 has been able to do, but without factory farms or unsustainable methods. So how does this work? What's really exciting is that we don't even have to harm the animal to source the stem cells. Do you know that fresh milk has viable stem cells that we can isolate? We're able to isolate these stem cells, expand them to a large number, and that's when the real magic happens. When exposed to lactation media, these cells differentiate into memory cells and begin to express milk. What's even more amazing is that these functional cells can continue expressing milk for up to 200 days. Now that we have this ability to create something so precious without harming the environment or animals, what can we do with it? Do you know that all infant formula globally uses bovine milk powder, and that 45 billion market is going to double over the next 5 years? We're able to address that and the existing $700 billion dairy industry that uses raw milk as a starting point. Very early on, we understood that if we wanted to make a global impact, we need to work with existing dairy conglomerates. The fact is they don't hate the planet or disregard animal welfare. They just need real solutions like TurtleTree to continue providing their customers with the best products. In the dairy industry, today there are 3 main layers. We have the consumer brands like Nestle and Danone, the dairy producers like Fonterra and Dairy Farmers of America, and equipment manufacturers like Gia and Tetra Pak. All of them make it possible to run the billion-dollar plants that support milk for the world. TurtleTree Labs is focused on a B2B model, working with all these partners to make it happen. Here's a brief history of TurtleTree Labs. We started the company in January 2019 with several scientists. We filed our provisional patents after we made some breakthroughs. In Q3, we started collaboration with Singapore's A-Star National Research Institute and were featured in many prominent publications. In Q4, we raised funding from major global investors like Level VC, Artisan, KBW, and K2 Global. Fast forward to today in 2020, we have 6 fully built out teams and have began customer engagement with major brands. Here's our executive team, and it's a good combination of seasoned business and science experts. And this is really a great exposure for us as we continue to onboard world-class talent and raise funds. We want to highlight the importance of food security and the need to build solutions to feed the next billion people sustainably. TurtleTree Labs will be a major part of the solution, and we thank you all for supporting us.
Thanks, Fengru. That was great.
Thank you.
I was really looking forward to seeing this. And I'm curious, just to kick things off here, what kind of hurdles have you gone through to get this to market, meaning being able to work with these food companies? I'm assuming, I know the US market obviously a little bit better than I would know, in this case, Singaporean. Can you talk to us about that and how it's regulated?
Sure. So one of the biggest challenges as a startup or any food tech startup is really scaling up, the scale-up process, which is why we strategically place ourselves here in Singapore. We have a lot of government support. There's the National A-Star Research Institute, whom we're working with to build out the large-scale bioreactors that we can scale. From a 5-liter, we're able to scale up to 50,000 liters. And these are some of the support that we've been getting internally within Singapore.
Got it. Ella, do you want to take the next question?
Yeah. So what? I know you talk about the B2B model, and your website talks about licensing and royalties and consulting. Why not consider going direct to consumer when so much of the volumes are going to produce initially are arguably going to be small, given the total size of the market, and the margins and the unit economics and just your ability to control your destiny in those initial months and years. It just tends to be could be a friendlier way to launch. I would love to hear your thoughts on that.
Sure. So this is a really good question because TurtleTree Labs, we are really mission focused, we're mission driven. And right from the start, we wanted to make a difference, we want to make an impact in the world. And I came from Google, and we have a big focus on a 10x effect. So in order to have a 10x effect on the technology that we're building, we need to be able to work with partners to drop our technology in different parts of the world. If we are to set up a brand, we have seen dairy companies or manufacturers, each of these plants cost $800 million. And this is not something that startups like us can afford right off the bat. So we do want to work with industry to help them to migrate to our method of sourcing milk. And this is a way that we can create a larger impact. And not to mention, they also have a lot of experience with go-to-market strategies, with existing logistic chains, cold chains. So we want to be able to tap on their knowledge as well. And this collaboration has been very well received, actually. 4 out of 5 largest dairy companies are having discussions with us. So it's a good path for us to scale.
Yeah, thank you. I think the risk, it's interesting how you're thinking about the risk, because I do agree. The capital requirements for independent scale up are sizable and scary, arguably. But if you think of first of all, you can always hire people from that industry at later stage in the company. So I think the experience and the knowledge, you can just hire people from those companies who would be really valuable for the company. I think, and you can always raise money, which I know doesn't sound easy, and it's not easy, especially at the rounds, right? Be it venture capital or venture debt or alternative funding instruments that make sense. But there's this interesting aspect of controlling your own destiny. And I think as many startups have learned the lesson. So it seems like you're smart about it, that you're talking, to more than one, and talking to 4 of them at the same time actually can, I think, create some really interesting competitive dynamics. But it's sometimes those conversations end up slowing down, and the perceived risk in the bigger companies is not very well aligned with the pace of innovation at the startup, or like how quickly a startup should move. So it's an interesting challenge to manage. I think there's an interesting question from the audience that maybe you can ask right now, and before we move on to Ashok. It's the top upvoted one. What can be used as raw material for converting cell-based milk? Have you considered greens to create milk with a nutritional profile resembling grass-fed dairy?
Sorry, can you repeat that question again?
Yeah, essentially, what can be used as raw material for converting cell-based milk? So I'm assuming some additions to growth medium. And have you considered greens to create milk with nutritional profile resembling grass-fed dairy? Does it make sense?
Yes, it does now. So it's interesting because it's a good question. We do have 2 types of media that we are working with. And the first media is a media that helps our cells to proliferate, to grow to a large enough number. And we are using plant-based options. We are not sure if that affects the taste of the milk, because that really just helps the cells to grow and multiply. The second lactation media is what's interesting. It's made up of the same components as inside a cow udder or a human breast. So the items inside are things like vitamins and minerals and some hormones, and it doesn't affect the milk that comes out. In fact, the milk that comes out of our bioreactors is actually quite neutral tasting. The grass and the grassy taste would need to come from the gut, the microbiome, and a lot of the other components that would be present in the milk.
Interesting.
I hope that answers that question.
It does. I would say that was my next question, which is what is the media you're using? As you know, in lab rat meat, there's a big debate around fetal bovine serum. So I was just curious how you were going about it. I think we're up on the 8-minute mark. Actually, we will probably have more time for questions at the end. So I'll go ahead and introduce Ashok. Thank you, Fengru.
Thank you.
So next up, we have Ashok from, I hope I'm pronouncing that right, from Stratuscent.
Hi, yeah, that's right. Yeah, Ashok. Yeah,
Great.
Thank you. All right. Hi, everyone. Good morning. This is Ashok Maslamani, and I'm the founder and CTO of Stratuscent, a company that builds smart nose technology, which we are trying to apply for accelerating the food supply chain transformation. Let me jump into the presentation. So what are we solving? Here, there is a graph that I actually pulled out from a report that presents, where food wastage occurs. What we observe here is food wastage, occurs in a number of locations. Mainly it looks like a lot of that occurs in, for example, the consumer location, in the food processing locations, retail and transport, etc. Food wastage represents in Canada, and this graph is from Canada. In Canada alone, back in 2014, it represented $31 billion in wastage, actually. But in general, across the world, food wastage represents about $1 trillion worth of material wasted every year. So what has happened is in Western countries like US and Canada, there are legislative goals to reduce food wastage and loss by 50% towards the end of this next decade. And to do this, you need a number of tools, actually, which needs to be applied in a very contextual way across the entire supply chain. And let me explain how we might be able to help with our technology. So what are the current state of solutions when it comes to look at quality of food or looking for wastage in food, right? So obviously, we all know there is visual inspection based on cameras or even humans looking for produce or processed food that are essentially going through a conveyor belt. But the issue here is there is limitation in terms of what you can identify, because visual cues are very limited when it comes to food. More comprehensive ones to control quality or look for wastage could be lab testing, but lab testing is costly, non-real time. You have to take a sample, go to the lab and test it, and the throughput is very low. And there are certain cases we actually talk to various processors where humans are used as sniffers to look for indicators from smell, which is one of the important indicators of food spoilage, because there are pre-spoilage chemicals that gives information way ahead of time about spoilage that could happen. But the problem is humans are very subjective, and also the throughput could be a lot low because you may not be able to deploy it at every key points to monitor for the spoilages and extra. So what have we done here is at Stratuscent we have built basically a replacement for a human olfaction system that can even work better than the human olfaction system. But what we have done is, instead of creating a solution from ground up, we try to mimic how our olfaction system works, which actually is a pretty impressive system. Our olfaction system has an array of neural receptors that produces a neural fingerprint every time when we take a sniff of air, and depending on what kind of smell or chemical that is present in the air. And that neural fingerprint is very unique to what you're smelling, and then the brain actually processes that fingerprint and does a classification of oh, I smell coffee with a background of pizza, etc. In the same way, we have put together a sensor where we have an array of chemical receptors that react to any smell or chemical that it is introduced to, and then produces a unique, what we call internally as a scent print, which is very unique to what it is smelling. And that scent print is basically sent to our cloud-based centralized AI, which acts like a brain and performs, classification of, okay, what I'm smelling is, this particular smell, even if there is a complex background that is there, right? So it has the ability to subtract for complex backgrounds, variation in variables like temperature and humidity, and it also can actually track the evolution of any complex smell. So it's not just like one snapshot. And we can do all of this, at a low cost in a device that is very modular and basically aiming to be deployed at large scale for a scalable solution essentially. Just to give an idea, how does this device look like? Before jumping into that, the technology itself, we actually started with a prominent lab, so from Jet Propulsion Laboratory at NASA. So they had developed electronic nose technology, and they had filed 6 patents around it. We actually in-licensed exclusive worldwide rights to those patents, and so that's where we started this work. We have further developed the technology and have produced a miniature modular device that can be quickly adopted for various form factors, right? You can use it in a handheld manner for a point and shoot kind of application where we can observe what is happening at a particular location, or we can put it in a wall mount in any kind of facility. Here there is a picture of a device that is about 4 by 5 centimeters square, and it's mounted onto the hood for monitoring certain processes here. As a connected device, it has a centralized AI access, which actually can avoid false positives, because every device that goes onto the field feeds into the AI, which means they do a lot of cross learning and has an exponential learning capability of unseen environments where it can operate. In terms of response time, the sensor itself responds under 5 seconds, and the interpretation is less than 10 seconds. So it's really fast. It's quite real time in terms of what it can do. So what can it do with a device like this? One can imagine, can we detect, for example, pre-spoilage chemicals across the entire supply chain, from storage facilities for produce to transportation trucks to retailers all the way to consumers? Can we, for example, help with controlled ripening? Can really avoid spoilage and actually, sell the ripened food at the right, stage for the most point for consumers, actually, where we don't lose the nutritional value. Or can we do pick the best harvest times based on the aroma profiles for the growers, etc. So we believe we can bring a lot of value across the entire food supply chain if there is a low-cost device that can use smell as a vector. So in that sense,
I'm here to give you a warning.
Okay, yeah, I'm almost close. So there are two example use cases I wanted to highlight. We actually have a number of customer engagements. One of them is in raw milk quality assessment with a dairy processor in Canada, where what we have done is we have already proven the technology that it works for the problem. We are able to separate the quality of raw milk from good raw milk to 8 different bad spoiled raw milk, and it is very useful for them in terms of incoming raw milk, and as they take it to further processing to avoid wastage of milk, as well as improve the quality across the entire processing supply chain. And that is in the case of a processor. We also have, for example, a hop growing company that wants to use our sensor to scent print the aroma profiles of their hops, which can be used for producing consistent blends for the beer industry, where we have started early testing for them. So those are some of the use cases we are already working with and one of the two of the various customers that we are working with. So where we have identified various target markets in the food and beverages industry. We have some engaged partners. We have prototypes deployed and proven the technology for some use cases, and in some cases we have field deployment going on. We're looking at horizontal market scaling for various use cases, etc. So we are looking for customers that want to partner with us to develop solutions for the pain point. We're also looking for investors who are interested in investing in a groundbreaking transformational technology like digitizing the world of smells. Here is my contact. Thanks for listening to my presentation. Thank you.
Thank you, Ashok. That was great. Really interesting. I've been was reading your deck earlier. I've got so many questions. I don't even know where to start. One that came up. One thing I'm curious about is why. I understand that the food industry uses smell as a way to detect spoilage, but in your research, how did you decide that to go after smell versus testing bacteria, for example?
So it was actually a reverse problem in our case. We started with the technology because we truly wanted to digitize the world of smells. Where what we have done all the smart devices is we have digitized the world of vision, where your phone can detect your face, right? It can talk to you, it can listen to you, it can sense your touch, etc. Right? To smell, we have literally nothing in our smart devices. So that's where the entire idea started, and as it started evolving, when the technology was developed, naturally our early customers who really wanted to use smell as a vector to solve problems were always in the food industry, which is where it was a lot of inbound interest that really wanted to tap onto a variable that they never had access to. Because it opens a completely new world for them, and that's how we essentially latched onto this.
Cool. Got it. Ella, you want to take the question?
Yeah. So how? So I looked I looked into the eNose research, and it seems like the research has started around 30 years ago, and there's multiple eNose approaches. There are protein-based approaches, chemical, conducting polymer sensors. How did you decide that, first of all, with licensing from licensing the technology and also the development, how did you decide that this is, and what gives you the confidence that this is the right technology to focus on and continue development on for those applications? I'm assuming all of them have different applications, but how does that compare to other eNose technologies?
Yeah, you are right. The eNose industry is 30 years old, and it's littered with a lot of what we call as failed eNose startups actually, and not because they did anything wrong. Many times actually the technology couldn't cross over a barrier at that time, in the 90s, in the early 2000s, etc. So there were a number of problems. There were 3 key problems that I identified when we started this company, back in 2014 summer, where the idea was getting concocted. The first document that was written was not actually business plan. It was actually analysis of the companies that failed before and what we learned from it. It's a document that is there in the company that everybody goes through. One of the problem was scalability of manufacturing of the sensor itself, which was a huge issue because most of this, electronic nose sensors, they don't fit into the standard CMOS or MEMS regime for scalable manufacturing. What we have figured out was, after the breakthrough with material printing, due to the 3D printing wave in 2014, we have actually ways to produce scalable sensors through printing technology. So that got solved. We also needed AI that was capable of actually look for learning from various devices, across multiple sensors and do cross learning, and can truly subtract complex backgrounds to interpret what smell it is actually currently smelling. To do that, you needed a computational platform that was powerful enough, right? And again, if you go 2014, the cloud computing surge that happened at that time enabled such capability, and we also had access to novel algorithms, right? Because the big push in AI happened where approaches that were not considered before started getting implemented into. Took advantage of that, but at the same time, we also realized NASA had done some good work. For example, in their patents, one of their patents filed for us, they were able to unmix 2 different chemical signatures in an open environment. And they were the first to do that, actually, because it is a lot easier to do it in a controlled environment in a lab, but in an open environment with the presence of background humidity, temperature variation, etc. And that was the key. We actually understood how they approached processing chemical data, which was very different from everybody that had done that before, right? And kickstarted from that approach, took it to a next level, moved from just simple chemicals to actually unmixing complex smells from complex backgrounds, etc. Right? So we have a robust set of IP that we filed around it, beyond what even NASA did, so that's how we have differentiated ourselves in a meaningful way.
Cool, thank you. And I would just say, I'm happy that it seems like from looking at your website, it seems like you have this very broad list of applications there. And I think, especially knowing that there are different technologies out there, I think it's, first of all, hard to be the best at all sorts of sensing. And then it's very hard to do business development in all those directions, because it's, so I'm really happy that you're focusing on it with the initial pilots on food and beverage, because it's just very hard to build a company pursuing very different targets when it comes to customer profile and types of molecules. Great. I think we will leave it here, and then there's going to be a general Q&A. Thank you so much for the presentation.
Thank you for the opportunity. Thank you.
Yeah, I think I'll go ahead and drop off and let you take it to the group. I'm sure there's lots of questions in there.
Great, that was fun.
Thanks, guys.
Thank you.
Okay, so I think actually Ashok, Fengru, and Steven, I think you should all join me now. Awesome, you are all here. Great, so let's look at the questions. The top. So I think it's the question, we'll start with you, Fengru. So if you want to unmute yourself, how will you separate the complexity of milk molecules and nutritional components of your product from the media you're using?
Sure, so we are doing that through the special bioreactors that we're using. The bioreactors that we're using are called hollow fiber bioreactors. So what happens is, if you think of a giant tank, and inside the tank there are hundreds of straws. So inside the tank is where the lactation media sits, and the cells are adherent to the outside of the straws, and the straws are porous, so it's full of holes, right? So what happens is the lactation media would pass through the cells. The cells would convert lactation media into milk and pass through the inside of the straw to an outlet. So this filtering process is very much used for human insulin production today, as well as wastewater management, which is why it can be used to scale up to 50,000 liter bioreactors. And they're linearly scalable because the air bubbles can be quite uniformly channeled through the bottom and the top of the bioreactor.
So you're saying that the cells are secreting milk continuously, and like you never, when you harvest milk, you actually don't touch the cells. The cells kind of, okay.
Exactly, and it should be a closed loop environment. It should be aseptic because the cells are quite delicate, so we don't want to expose them to the air. So this environment is all closed loop, and it's a continual flow process. So our cells act as the factories for creating milk for up to 200 days before we change out the cells.
Great. So another question, which I think is a general question, if we can all talk about it. So plant-based protein is known to have less protein quality, and that's in your quote, air quote, sorry, i.e. less digestibility and less proportion of essential amino acids in comparison to animal protein. Is this an issue for new plant-based food? That's the first part of the question. And what is the role of new sustainable alternatives like insect protein? That's, if you guys have opinions, we can talk about. Okay, so I'm actually not an expert on insect proteins bioavailability. So if there was an intention that I would be able to comment here, I'm probably not the one to add to shine too much light on that. You know what? I think the next there's a. Okay, I think there's a general question for all of you, which well, maybe let's start with Steven. How to, and I'm sure it's kind of intended, I'm not sure which company's intended to, but how to address questions of regulatory approval. So kind of one by one, if there is any approval process when, I guess when you're ready to introduce prebiotics and symbiotics.
For us, it's a little complicated because each type of product has a little bit of a different path. So we're focused on, from a food perspective, on grass and achieving grass status and going through the relevant safety trials. Obviously, therapeutic applications and IND applications are a different path altogether. So for us, it really is dependent on the product and the product profile in terms of how we proceed. But yeah, it's always a complicated topic.
And then, Steve, would you be working with partners to go through approval, or I understand these are secondary questions and maybe later down the road, but do you see that happening in-house where you, when you bring certain targets to the market, or would you at that point license some of the targets out or work with partners?
Yeah, so we will always be a B2B company. We don't see ourselves as eventually marketing. I think it's likely that with food products that we'll take on more of the responsibility of safety trials and the regulatory aspects, and before we partner those ingredients out with therapeutic applications, it's harder to say. But we will, at some point in the process of bringing oligosaccharides to market, through whatever path, we'll be partnering with the marketing company. But for foods, we intend to do the safety trials and the clinical trials ourselves.
Cool. So Ashok, I don't know, maybe same question to you, and it's in terms of any regulatory approval. I'm assuming for the initial pilots, that's not, it's just kind of you proving efficacy, but tell me how, are there applications where regulatory approval will be needed, and how do you think about that?
Yeah, we have thought about it, and one of the approaches is, since we work with actual partners, they actually are integrating our solution into their facility and solution. So we get the secondary list of things because they go through the approval process on behalf of us to introduce a new technology. So we work with the partners to meet those requirements that are laid out for us. So usually it's a partnership that approaches it, right? For example, even in the dairy industry, whatever we are building right now, we already, like the dairy processing company, are trying to work with the milk boards across, like, the Quebec Milk Board, the Ontario Milk Board, and all other milk boards to introduce this, to them ahead of time, so that we can actually be ready for the regulatory set of requirements, and we'll be actually meaningfully address them on time. So that's one of the approaches we are going through in a collaborative way.
Cool, guys, we have 4 minutes, and it's soon going to change to 3, I'm sure. So there's 3 of you. Let's use it to, if you can, it's one of the questions we love to ask at 50 Years is, this big vision question, you know, like kind of why do you wake up every day to do what you're doing? And like, what was the initial, and what is the most happy outcome of what you're doing? So if you can imagine it's 10 years from now, and everything went exactly as planned. Like there hasn't been a single challenge. It's this hyper optimistic scenario. You know, everything. In fact, everything went beyond the plan. So like, if you can put yourself in that mindset, you know, what have you achieved, and how is the world different?
Sure, I'll start. So for us at TurtleTree, we really want to change the world. So in the next 10 years, 7 years, in fact, we want to see TurtleTree technology in every carton of milk that is being produced by all the major dairy manufacturers. So we envision our logo to be at all the cartons. So how we model our business model is after a company called ARM Semiconductors. They're the largest chip company in the world that doesn't make a single chip. So they work with Samsung, they work with Apple to help them to transform to their own technology. And they have grown tremendously, and they became a shared resource of the whole tech industry to build IP around digital architecture. So this is what we want to be. We want to be that technology that allows dairy companies to continue accessing sustainable dairy for their customers, quality products.
Great. So TurtleTree inside.
Exactly.
Great. Steve, do you want to go next? 10
Yeah,
years.
Sure. Can you hear me? We've got two real opportunities for benefits for society, I think. The first is improvement of health, improvement of chronic disease conditions. And the other is the reduction of waste in the food supply. So carbohydrates are the largest source of waste and waste products. And the technology has the ability to connect these two benefits with a single solution, you know, bringing that waste stream back into the food supply and converting it into a form that actually provides long-term benefit for preventing chronic diseases in humans. So I think the ability to connect those two benefits together with this technology is where I would really like to see us end up.
I love it. I hope you make you have a massive impact there. Ashok, you're the last one. 10 years, hyper optimistic scenario.
Yeah. What we see as is like if you look at Google, you know, they've created a tool which is the search engine that enabled them to.
Oh, and you froze. Can you hear us? We cannot hear you, but from all you were able to say, I'm pretty sure you're saying you are the Google for smells. So you're the search, and I'm just hopefully doing justice to your big vision, as we cannot hear you. But it is an exciting vision if I'm getting it right. Yeah. Okay, guys, this was unfortunately we lost them, but I think it was Google for smells, which I think it would be an incredible vision. I'm sorry we lost the Ashok. We are almost done, I believe. Are there any questions that I guess we should probably Oli and the team at Rethink, do you want to step in and say the final goodbyes? If you can hear me. I see a lot of, I see many positive comments on the presentation quality. I thought they were phenomenal. I think there's so much to learn, and actually it was very inspiring to see that, you know, I think food and food tech often get, underappreciated with like how much science and how much complexity there is and how much innovation we need to really move the world forward. So, I mean, everyone's work here is incredibly impressive and exactly the types of solutions we need at scale to really make a difference to the many food and environmental problems and animal agriculture problems we have. So this was really inspiring to me.
Thanks, Ella. Thanks for hosting.
Yeah, thank you so much, guys. Firstly, thank you so much to the presenters, Feng Yu, Steve and Ashok. I'm really sorry about the technical issues, and thank you, Ella and Holly, as well, for making sure that it runs as smoothly as humanly possible. And but yeah, I want to echo those thoughts, how amazing those presentations were, how in depth and exciting those solutions are, and really looking forward to seeing the next 10 years and how that's going to transform the food system. So once again, yeah, thank you very much, guys, for joining. Thank you all to the audience for submitting those questions. And our next webinar event will be next month, and that will be on consumer trends driving ingredient innovation. So please do register for that. And we've also launched our new virtual summit as well, which will be taking place in the middle of September. And you can find more information about that on futurefoodtech.com. So yeah, once again, thank you very much, guys, and enjoy the rest of your day.
Thank you. Thanks, everyone. Have a great day.
Thank you. See ya.